Hello everyone, and welcome to our final episode of Chapter 18. We're wrapping up the fourth lecture here with the second part, and this is one of the tougher concepts, so we're going to be doing some application here, and I saved this for the end. The publisher, in fact, actually starts with it, and I understand why they start with it, because of course they're talking about how hormones function, and so it kind of makes sense to start early on with showing how these water-soluble versus lipid-soluble hormones function. I, however, prefer to give students a background knowledge on what hormones do, because I kind of find that a little bit more interesting, a little bit easier to follow, and then close out with the functionality. So just a minor switch in comparison to the book. So if you were reading the book and you're like, wow, this was introduced and Mr. C hasn't been talking about it, I saved it for the end here. All right, so let's get into this. As you can see, part 2, we're really just focused on the lipid soluble versus water soluble. So really understand these objectives. They are super important here to really note. But as usual, I don't cover these. Feel free to pause it here if you want, but we're going to get into the lecture content. And before we get too deep into the new content, we got to review some concepts. If you remember, we started talking about free versus bound hormones. That information was kind of scattered throughout the lecture, so I want to kind of hint at these and be a little bit more specific on these. So when we talk free hormones, remember we're talking, these are going to be the hormones that are released into circulation that do not need to be attached to a protein. So we say they're unattached to a protein. This is abbreviation for circulation if you didn't know that. All right, but notice these hormones that we're talking about, these are going to be our water-soluble hormones. And at this point, you should be able to recognize what are water-soluble hormones. I've been trying to stress that throughout all my lectures and all the episodes. And what we, of course, are recognizing is these are the peptide derivatives. So since they're composed of many amino acids, we know that, typically speaking, proteins in general don't cross the bilayer. They tend to be polar. They tend to be large. And therefore, they're not going to just diffuse through a bilayer. And so that's going to be peptide derivatives. We have the catecholamine, which were, of course, the tyrosine derivatives, epinephrine, norepinephrine. And then we talked about the tryptophan derivatives, which those, again, are primarily the amino acid derivatives with the exception of thyroid hormone, because, of course, thyroid hormone is going to be a bound hormone, or we know it's going to be lipid soluble, right? But expanding on this definition here, these are going to be hormones that remain functional for less than an hour. And the reason for this is they're not two molecules. They're just circulating in space, and therefore enzymes can actually attack them right off the bat without having to attack two different things. Where bound hormones, when we talk about them, they're going to be their hormone itself and an attached protein. So enzymes aren't going to be able to function on those directly. And so these remain functional for less than an hour, i.e. they get broken down quicker than your steroid derivative or your lipid-soluble hormones. Now remember, bound hormones, these are hormones that circulate attached to a transport protein. And as I was saying, they are harder to break down because they're two molecules. They're a steroid molecule that is attached to a protein. So technically you need two enzymes to break them apart. And because of that, these are going to remain functional for longer periods. We don't have enzymes that can easily access them and break them down, and they're going to just remain in circulation longer. You might have heard of water-soluble and fat-soluble vitamins. The fat-soluble vitamins, although harder to get access to, they're going to get stored a bit longer, where water-soluble basically runs through the body. So keep this in mind. This is a concept about water-soluble and lipid-soluble, and understanding which are going to actually survive longer periods of time. But regardless of the type, there's two processes that basically occur with these hormones as they're circulating. One, they bind to the receptors on their target cells, which we call the effectors, and that ends up leading to a response or causing those target cells, to perform its specific function. So whatever the hormone is going to trigger, basically bind to the target cell and cause that function. The other option is they're going to be broken down. Now, we can break them down where they get recycled by the liver, or they can be completely removed by the kidneys. So oftentimes we're going to learn that the liver breaks things down, processes it, and then sends kind of the remaining waste product to the kidneys. We're going to see this in later lectures. We're going to talk about the recycling of red blood cells, and we're going to see the liver breaks it down and then sends the remaining waste to the kidneys. So understand, it can be broken down, or it can actually be destroyed by enzymes in the plasma or interstitial fluid. And that's what I was talking about earlier, why it's easier to break down free hormones, because it's literally just a single hormone or a single molecule that can be attacked by the enzyme. And so think something like acetylcholine esterase, if you remember talking about that when we were doing muscular contractions. This is what broke down acetylcholine to stop the actual signal of muscle contraction, right? So that's kind of what you can think about related to this to have kind of a good understanding here. Now, we're going to move on to the part, and we're going to talk about which hormones bind intracellular receptors. And, of course, you should be thinking about this. You should actually know the answer. We've talked about it a few times, but pause it if you want to think about it, or if you're paying attention and you just look at the image. Obviously, the image is the one that does. And as you can see, it's going to be our steroid hormones and our thyroid hormones. So we're going to start with steroid or our lipid-soluble hormones in general, and talk about how those hormones function, because it's a little bit, it's pretty straightforward. There's no sending the message between other molecules. It's literally what happens with these is they go through the cell membrane, as you can see here. All right, so what would actually happen is, which isn't really shown, they would of course have to unbind their transport protein, but the main point is we're focused on them, how they're going to function on the cell. And so notice they're going to pass through the cell bilayer, and they're going to bind to an intracellular receptor. And when they do that, as far as steroid hormones, and that's what a steroid looks like, that's the common chemical makeup of a steroid, so you should recognize that that's a steroid. Anytime you see this four-membered ring where it's basically three hexagons with one pentagon attached to it, that's always what represents a steroid. And what they basically do is they end up binding an intracellular receptor, and when they do that, that leads to protein synthesis. All right, so notice binding to a receptor, gets brought inside, activates DNA and causes gene activation, which leads to the production of a protein. So that's literally what steroid hormones do. They bind to a receptor and they start protein synthesis inside that specific cell. That's what you need to know. You need to know steroid hormones bind intracellular receptors, and when they bind the intracellular receptor, it leads to protein synthesis or gene activation, however you want to phrase it, but that is the main point for steroid hormones. Again, your steroid hormones are basically your testosterone, your progesterone, your estrogen. Those are your sex-based hormones, and those, of course, are secreted by the gonads. You have cortisol, right? And so cortisol was one of those major ones. That was the glucocorticoids. You had aldosterone, both of these being secreted in, of course, the adrenal cortex. And then generically speaking, you can list the androgens, right? The only other thing that's missing on there would be calcitriol. So if you wanted to add, of course, androgens and the calcitriol, that would cover your complete list of steroid hormones. So it's not on my notes. I would actually add it to yours to make sure you got those down. Calcitriol, and again, androgens. And that makes up all the steroid hormones. Of course, thyroid hormone is also a lipid-soluble hormone. Make sure you understand it's lipid-soluble, but it is not a steroid derivative. This hormone can also activate genes, and it essentially controls the rate of transcription, but notice it can also bind to a membrane on the mitochondria. Remember, the mitochondria is made up of membranes. And so notice option B is it can actually stimulate ATP production, and that's how it really regulates metabolism. All right, so no thyroid hormone. Understand that that has two functionalities. Bind to intracellular receptors, and in some cases leads to an increase in ATP production, or also leads to, notice, protein synthesis. So get these down. Thyroid hormone, we spent a lot of time on it individually. You really need to know this hormone really well. Notice targets most cells, important for cell development or just in development in general. All right, but overall, this should be the simple one to really understand. So make sure you work through this and have this well understood. And really the main point from this is lipid-soluble hormones function by binding to intracellular receptors. And that's what we're seeing on both of these images. They're binding to intracellular receptors, however we see it, which often results in the gene activation, which you should know. Gene activation means we're leading to protein synthesis. This statement should make a lot of sense, and you should get to this point feeling comfortable with, hey, I know exactly what this means. Now, regurgitating the statement isn't what I'm saying. You need to understand what the statement means, because what that really implies is you understand gene activation means protein synthesis. That's the connection you need to have here, and you really just need to know lipid-soluble hormones pass the bilayer and bind to intracellular receptors. When they do so, it often leads to gene activation. In the case of thyroid hormone, it can lead to increased ATP production. That last statement there was the full summary that you needed to walk away with. It's going to take a bit to work through this. Really focus on these images. Pearson does amazing jobs with these images, so you need to spend a lot of time reviewing them, and they should make sense to you. Once you understand the concept, this entire image will make a lot of sense. Now, of course, they're talking about binding to DNA and going through transcription and talking about mRNA. You don't need to know those additional steps. You just need to know it leads to the production of certain proteins. Notice Those proteins can alter the cellular structure or activity. All right. Now, that one was the easier of the combination. Now, when we look at the hormones that bind to extracellular receptors, this is where some challenging concepts start to emerge. And the reason I say challenging concepts start to emerge is, if you're thinking about this, it should make sense that this cannot be the only chemical, because we're not going to be able to get gene activation inside the nucleus by one chemical binding to the outer receptor. That shouldn't really make sense to you as I say that. Like, oh yeah, I guess that really doesn't work. All right, and so what we're talking about, the hormones that bind extracellular receptors are going to be any of the water-soluble hormones, which primarily you should be thinking of peptide hormones. Okay, and that's pretty much, if you knew the list back here, that's why I was saying write down that calcitriol on there, write down androgens, and you have your complete list. Everything else, except, again, thyroid hormone would be the exception as well, but everything else is going to be related to mostly peptide hormones. With the catecholamines, that's epinephrine, norepinephrine, and then tryptophan would have been your melatonin and dopamine, right? But why do these types of hormones have to bind extracellular receptor? What makes something water soluble? And hopefully you understand that these are not lipid soluble, and the reason for it is they're large. So peptide hormones, they're large. They're polar. That polarity is what really ruins it. Lipid-soluble substances are nonpolar, and so all of these are going to be polar molecules, and they're hydrophilic. So the polarity makes them hydrophilic, meaning they love water, where the nonpolar molecules are going to be hydrophobic. All right, but in this case, what complicates the matter is we have to actually have two messengers. Now, there's actually going to be a whole bunch of chemicals inside the cell, all right, but the first messenger is going to be the hormone itself. It's the indirect response. So note, I would circle this and write off the side or make it very clear that you understand that the first messenger is going to be your water-soluble molecule, which mostly is going to be the peptide hormones. Again, catecholamines still fall in here. The tryptophan derivatives also fall in there, but I'm going to stress the peptide hormones because that's usually what I focus on in the exam, and I think it's easy for students to understand. Peptide is proteins. Proteins are large and hydrophilic. All right, but the first messenger is going to be the actual peptide hormone or the actual water-soluble hormone that binds to the plasma membrane receptor. So here's the plasma membrane. That's the outer portion of the cell, and it's binding to it. What you really need to know, and I test heavily on this, the first messenger, actually what it does is when it binds, it activates something called the G protein that they're showing down there. And the activation of the G protein is a chemical intermediate that's really responding to this first messenger, and it eventually leads to the activation of the second messenger. Now, again, there's going to be something called a chemical cascade that we're about to talk about it. But when it comes to secondary messengers or second messengers, understand that these are going to be cytoplasmic molecule. All right, notice cytoplasmic means inside the cytoplasm, so don't overthink a term like this and say, I don't know what cytoplasmic molecule is. You're just combining things that you should know. You should know what the cytoplasm is. That's the inside of the cell. So a cytoplasmic molecule is a molecule that's inside the cell itself. All right, but that's going to carry out the actual response. And as we can see, a lot of it's going to be activating enzymes. Activating enzymes are opening ion channels. Here we see one that's a reduced activity, and we're going to be talking about this. Like I said, this last little section is one of the more challenging concepts of the chapter here. All right, but ultimately, second messengers usually activate genes. In some cases, can increase and decrease metabolic activity. Think epinephrine, norepinephrine, right? And so, again, this first part, we're posing the question because we're going to reiterate the points. What does the first messenger do? When it binds, what does that lead to? You should be able to answer this top question. When a first messenger binds to its receptor, and you should know it's an extracellular receptor, that binding leads to the production of second messengers by activating G proteins. Okay, so that's the whole concept. A first messenger will activate a G protein. The activation of the G protein ultimately leads to the production of lots and lots of second messengers inside the cell. And these second messengers can be different from, target cell to target cell. If two hormones target different tissues, they can have different responses on the tissues. So keep that in mind. In fact, that's how the negative feedback loops ultimately lead and work, right? Because in negative feedback, usually your first messenger is going to end up causing a decrease of a secretion from that specific organ that's releasing the hormone. But we'll talk about that in a second. That statement might not completely make sense yet, but you'll see it in just a minute. Just think the negative feedback loop. We're going to hit our target cell. to cause them to produce lots of second messengers, but ultimately the first messenger will also target itself and usually decrease the production. All right, but what you have to understand about this first messenger is a lot of the response is going to be based on intracellular enzymes. So having enzymes inside the cell already ready to go, because we're going to talk about a chemical cascade where literally one molecule activates another, which activates another, which activates another. That's called a chemical cascade. But ultimately, if the intracellular enzymes are correct, we have what's called amplification, which is a huge response that can occur to really low hormone concentrations in general. And that's amplification. Again, I'm telling you how the cells, what the cells can do to kind of amplify a response off of a first messenger. So the cell that's being targeted, if it has the appropriate enzymes, will lead to amplification. Okay, so that's just the term what amplification is, big response to low hormone concentration. This is often seen with like luteinizing hormone, for instance, that leads to ovulation. Usually it's a small amount of luteinizing hormone, but it generates a big response. All right, and so cells influence responses by regulating their receptors as well. So they regulate the internal enzymes. They also regulate responses by regulating the numbers of receptors. So when we talk about these protein receptors, you can see this is a protein receptor. It's embedded in the cell membrane right here. These are not permanent residents. These receptors can be reduced in concentration or increased in concentration. In fact, we'll talk about that in, actually, I believe it's this one right here. So downregulation. So this is what a cell can do to modify the response. So downregulation is when a cell decreases the number of hormone receptors when hormone concentrations are really high. So if you have abnormally high concentrations and the cell needs a lower response, it'll break down some of these protein receptors. And since now we have less protein receptors, we're essentially getting less response. So that's what downregulation is. And all that really means, and what you should understand, is the cell has become less sensitive to the hormone because it has less receptors, which means less receptors, less gene activation, less of a response from the cells. That's what you need to walk away with this. Now, there is a disease that's characterized from a dramatic decrease in the sensitivity to the hormone. And maybe many of you know this. Can you think of a hormone that ultimately all of a sudden the cells become less responsive to it? Well, the disease, of course, and if you want to pause it, but the disease we're talking about is diabetes, specifically type 2 diabetes. That's, associated with diet, and there is a genetic factor to it, but it's often associated with diet. And basically your blood is sort of, you're resetting the range over time. And so basically there's so much insulin in the bloodstream that over time the cell says, hey, we need to decrease the number of receptors. There's just too much insulin. And so the cell will decrease the number of receptors, and now your body is not responding to insulin the way it should be. And that, of course, leads to higher blood glucose levels, which is what type 2 diabetes really is an inability to respond to insulin. It's we're going to find out it's called insulin resistant. All right, so people produce insulin, but the body is just not responding to it. Of course, if you have downregulation, there is the flip side. You can have upregulation, and that's what occurs when we increase the number of effector receptors due to a low hormone concentration. And so here, again, the cells become more sensitive. That's exactly what happens during ovulation, is the ovary increases the number of luteinizing hormone receptors, and over time, when now luteinizing hormone gets released, we get a much bigger dramatic response because of upregulation and amplification, which ultimately leads to ovulation. So kind of know these examples. These are mostly terms and definitions that I would expect students to be able to recognize here. So this is just saying what's happening is a first messenger binds, and it's not as simple as, okay, the chemical cascade occurs and we get a cellular response. The cell can control its response in some ways. That's all this slide is talking about, is how the cell can kind of modify its responses, and it's done through either downregulation, upregulation, or, again, do not overthink the one that I'm talking about, is the intracellular enzymes, having the appropriate ones there for the chemical cascade. This is hard material for me to test on, just so you know. To produce great questions with this is a little bit more challenging, but they are important premises that I think students should really understand, because I think it makes the other content a little easier to understand. All right, so we're going to look at several second messengers. And again, your key when we look at this is all right here. The hormone binds to the protein receptor. It activates the G protein. You need to know that. I guarantee you I'm going to test you on a G protein. To be clear, The G protein is a link between the first and second messenger. The G protein is not a second messenger. All right, this is not a second messenger. This activates the chemical cascade that leads to the production of second messengers. So what are the second messengers that we should be aware of? So here we go. Hormone binds, uses the G protein. G protein becomes activated, and then these are the events that happen. All right, so second messengers are cytoplasmic chemical messengers. They're often an enzymatic activator, can be an inhibitor, or in some cases can be a coenzyme, but ultimately the second messenger leads to a cellular response. That's what you need to get down. Second messengers ultimately carry out the final response. So the example here that we're going to start with, and there's only 3 second messengers we're going to talk about. These are the 3 most common. There are others, so if you take an advanced anatomy and physiology course that expands on these details, don't think that they're wrong. I am doing a very, very basic surface level explanation of this. There's a lot more details that anatomists know and physiologists know that we are not even discussing. So keep in mind, this is a very surface level, even though it seems super detailed. It's on the surface level of this content here. So G protein's activated, and notice what ultimately happens. There's some steps that they're showing that ends up leading to calcium coming in. All I expect you to know is that some peptide hormones lead to the release of calcium. All right, and it's done. First step, we activate the G protein. That's right here. Notice the G protein leads to these sort of molecules that we're not going to get into, but if you end up taking cell biology, or even I did research on cancer, and these are very common terms. IP3 was one of the main ones that I've studied. Doesn't matter. All you need to know is the activated G protein stimulates the opening of calcium ion channels or released via the intracellular components. If you remember, calcium gets stored in the endoplasmic reticulum, okay, and that's what they're talking about. So we can release it via the endoplasmic reticulum or actually open an ion channel for calcium. And that means calcium is going to diffuse in, or calcium is released from the SR, i.e., when a primary messenger binds and activates the G protein, calcium concentrations will increase, and when calcium increases, we lead to some sort of response. Now, you should know these. We've talked about 2 key ones from the previous semester. So if you remember, we ended with the nervous system. Remember that calcium going inside the cytoplasm is what stimulated neurotransmitter release, right? You should know that. You should remember that. That's how neurotransmitters are released. The action potential reaches and activated voltage-gated calcium channels. Well, when those voltage-gated calcium channels open, calcium went into the axon terminal to release the neurotransmitter. Similarly, the muscle contraction, right? We needed calcium. Calcium bind into troponin. Troponin moved tropomyosin out of the way, and that began the contractile cycle. So those are 2 things that we know calcium functioned, or at calcium levels carried out a function. So obviously calcium is important. This is why we talked about the hormones, parathyroid hormone and calcitonin, right? Because calcium regulation is key, because calcium is a second messenger. So for you, in the sake of this exam, for the endocrine system, you have to associate calcium binding to something called calmodulin. Okay, and what calmodulin is, this is what activates cytoplasmic enzymes, which is why I said early on it can be an enzymatic activator up there. So calcium binds to calmodulin. This activates cytoplasmic enzymes. Okay, and what that means when we talk about activating enzymes, that's activating a chemical cascade that, again, is going to lead to many different functions. All right, in this case, activating enzymes often is going to lead to the production of other proteins or modification of proteins to make them active. Okay, so your main point, activation of a G protein stimulates the intracellular levels of calcium. Calcium has many functions, but its main function is to activate calmodulin, and calmodulin will activate cytoplasmic enzymes that carry out the chemical cascade that ultimately leads to a cellular response. Again, I'm surprised on my notes I don't have the word chemical cascade. That was something that I was hoping to have, and I'm not seeing it on my notes. So off to the side, definitely write down, leads to the release of a chemical cascade, or the activation of a chemical cascade. And like I said, a chemical cascade is when one molecule activates another, which activates another. That is what calcium is ultimately going to do. It starts the chemical cascade to lead to cellular function. So think calcium is an activator molecule. It stimulates the cell. Okay, the other secondary messenger, now this is where things get unique. Again, I was trying to stress this. This is why it's really challenging. It's the cellular response is often based on the internal enzymes inside the cytoplasm. We are going to see cyclic AMP functioning in a unique way, okay? Ultimately leading to cAMP production. So cyclic AMP is cAMP. It acts as a second messenger, but we can actually cause the reduction of cAMP. So this is going to be a hormone that's inhibitory. All right, so cyclic AMP, it's an ATP derivative. AMP is adenosine monophosphate. ATP is adenosine triphosphate. So clearly they're linked, so they're a derivative. And it's going to alter cellular activity, again, based on intracellular concentration. So the higher the cAMP levels, the more responsive the cell is. Again, you probably should take this note. The higher the concentration, intracellular concentration of cAMP, the more response the cell is. Okay, so we have two options for cAMP. The activated G protein can increase, or it can actually lead to a decrease in the cAMP levels depending on the enzyme available. This is one enzyme, this is the other. And between arrows here, those represent the enzymes. And notice, G protein is activated. This is saying the enzyme that it activates and what it causes. Here, the same, this is still a peptide hormone binding to a different receptor. Maybe it's a different tissue. Notice, it's actually causing a breakdown of cAMP. All right, so excitatory effects increase the concentrations of cAMP, inhibitory effects decrease. I heavily test on this. I heavily test on G proteins. What are they? I heavily test on these enzymes, PDE and adenylate cyclase. You're going to need to really know these. Expect to see questions on them, ultimately understanding what a chemical cascade is. So cAMP increases, i.e. your cellular response will increase when the peptide hormone activates the G protein, which ends up activating the enzyme adenylate cyclase. So when you see this word, you should think increases cellular activity by increasing the concentrations of cAMP. More cAMP equals more activity. Get that somewhere on your notes. More cAMP, more activity. All right, so adenylate cyclase is the enzyme that literally will lead to the production of cAMP, and we see it right here. Increased production of cAMP. ATP gets broken down by adenylate cyclase and forms cAMP. cAMP ultimately leads to the opening of ion channels, just like we saw back there. So literally can lead to calcium coming in, or can literally activate enzymes by activating kinases. All right, so cAMP can activate a kinase. That's one option. This is an enzyme that phosphorylates. The word phosphorylate simply means activate. In fact, my research in cancer was all about how much phosphorylation was going on to a specific molecule. And basically, we were able to determine that by the thickness or the darkness of a band. And that's what I was studying, literally kinase activity, because more kinase activity or more phosphorylation meant more activity. And that's all this is showing you. It's a very key concept. It's going to be it's a hard concept to really understand unless you continue and move on to like research or more advanced courses. But from now, just understand cAMP works by activating a kinase that leads to the activation of other molecules, ultimately leading to increased cellular activity. The other option is for it to open ion channels, just like we saw with calcium. Okay, so same sort of concept. We go back, we look at this. Notice G protein causes the release or the opening of calcium ions. That's the main point there. Here, adenylate cyclase leads to the increase in cAMP, and by having cAMP, it either opens the ion channel or activates kinases, which activate enzymes, ultimately leading to activation of other molecules. Okay, and you can see, notice epinephrine, norepinephrine, in this case, leads to cellular activity. Notice in this case, leads to a decrease, because this is going to be the inhibitory side where we're breaking down cAMP. And just think epinephrine, norepinephrine for your heart and your breathing rate, but digestion, epinephrine, norepinephrine slow that down. So this is showing you the importance of intracellular enzymes on cellular activity. All right, the final slide, I believe, is this is the last one. So this is the decrease in cyclic AMP. Like I said, this was going to be the hard part here on this last episode. So when the G protein activates phosphodiesterase, abbreviated PDE, you should be thinking decrease in cellular activity. If I mention anything about inhibitory responses, it's phosphodiesterase. Anything about excitatory responses, it's going to be either cAMP and or calcium. All right, but with phosphodiesterase, if it gets activated, it's going to break cAMP down to AMP. And And because we have less CAMP, that means there's decreased enzymatic activity, which means a decreased cellular response. All right. And so the main points of CAMP, if you have high concentrations, you have more activity. Concentration is increased when the G protein activates adenylate cyclase. All right. First messengers, epinephrine, norepinephrine. You have calcitonin, PTH. You can see all of those kind of down there. And then notice low concentrations of CAMP lead to less activity. The decrease is occurring via the activation of phosphodiesterase, and notice epinephrine, norepinephrine represent that as well. Now, the thing that is important to understand, now I test on these definitely come up. You're probably going to see a couple questions on a quiz. You're probably going to see like 3 to 5 questions on the exam related to this. You just need to be able to recognize the functionality of these molecules. The main point is you see the enzyme phosphodiesterase, you know that is a reducing of CAMP. It breaks CAMP down, it causes a reduction. It is an inhibitory effect. Adenylate cyclase is an excitatory effect because it leads to increased CAMP, and when that happens, that can either open enzyme ion channels or it'll activate enzymes of a chemical cascade. If you go back and you look at the secondary messenger of calcium, calcium literally is going to activate enzymes, and that's it. That's all you need to know. What I've showed here, and the reason why this information is important, what's unique about this information, I know this was quite complex and I'm going on over 30 minutes, which I don't like doing, but the reason why this is so relevant is most cells are following this sort of methodology. Now, the things that change is the chemical cascade that we're not going into tons of details on. But those chemical cascades are ultimately what carry out the response, and that's what researchers study. But what's amazing about it is no matter the tissue, oftentimes you're seeing these same enzymes and these same responses. People that are studying a tissue and they see adenylyl cyclase, they're going to assume that that's going to be an activator. They see phosphodiesterase being activated and phosphorylated, they're going to know that, hey, that's referring to a reduction in activity. In many cases, I'm sure there's exceptions to the rule, but just understand that this figure right here I actually borrowed from a presentation that was given to us when I attended one of my professional developments. But basically what this is showing you, notice adenylate cyclase, it's active. There's ATP to CAMP. Step 3, inactive kinase becomes active. Step 5, inactive enzyme becomes an active enzyme, and it ultimately leads to a response. This literally is the response. This was a generic thing. I just typed in adenylyl cyclase. This is not mentioning the same book hormone. It's just showing you this is the chemical cascade. And notice target cell secretes Z. So that enzyme ended up leading to the final production and the secretion. This really shows you the overall response, where here, although it says activate enzymes, just understand the final response is actually production of something or increased cellular activity. Now, the reason these are important is drug companies take advantage of this and kind of study the molecules, the hormones that would activate these, and they try to make medications that would basically be able to bind to these receptors and possibly do the opposite. So trying to maybe trigger, if you want an inhibitory, maybe it's pain and you want to reduce pain, well, you're going to want this. You're going to want this sort of effect happening because that means pain is often associated with cellular activity, so you want to reduce it. That's how this stuff is used. Obviously, we don't have the time to break all of that down. I hope you're kind of like, hey, that's sort of interesting, super challenging. So usually challenging things scare students away, and I don't want you to be scared away. I just want you to understand these are the basic initial steps that go on to really understanding things at the cellular level. Now, that's very, very challenging. We just looked at the cellular level. Most of the lectures during Chapter 18 were looking at more of an organ level and big picture responses, which is why I saved this for the very end. Now, to be clear, you definitely need to know each of the 3 secondary messengers and what they do, and you also need to know what a G protein is. Make sure you understand the G protein is not a second messenger. It's what activates typically a chemical cascade that results in the presence of second messengers. All right, that wraps this whole lecture up. We are done with Chapter 18. Congratulations through making it through one of the hardest chapters. Next chapter is probably equally as challenging, Chapter 22. That's where we're moving on to. I don't move on to Chapter 19. 19 begins blood, and then we get into Chapter 20, the heart, and then we get into Chapter 21, blood vessels. All of that's the cardiovascular system, so I wrap all that together in Unit 2. So we're going to close this unit out by getting into the immune system. Those of you that are from another class, you might want to just jump over to chapter 19. That's going to be kind of out of order as I lecture on it, because I'm preparing most of these lectures for my class. But those of you that are kind of piggybacking and watching this, just keep in mind you might need to jump to chapter 19, and some of the concepts that I say that I direct at my students might be slightly different. Anyway, we're going to end here for today. Look for chapter 22. That's going to close out unit 2. So we're almost done with, unit 1. We're almost done with unit 1. We got to get through chapter 22, and that'll complete the first unit here, which will give us a lot to test on. If you haven't been keeping up with the lab lectures, make sure you do that, because the lab lecture stuff, the lab quizzes and lab exams are going to be right around the corner. So keep that in mind. You want to keep up with lab stuff because we tend to take those exams first, and then you take the lecture side. So you have a little bit of wiggle room with the lecture exam, but things are coming down the wire quick here. I know it seems like we just finished one chapter. How can we be at an exam already? But it's coming quick. Stay on top of this semester. It goes fast. Take care, everyone, and I'll see you over at chapter 22. Don't hesitate to ask questions.